Hello, my dear students. Welcome you all to Shri Jaindra Rank Zone classes. I'm a mentor and your friend for the subject biology, and I will be taking the zoology part, that is animal science. My dear students, in the last lecture, we have learned about the polymerase chain reaction and the applications of polymerase chain reaction. And now we are going to continue with the same topic. The fourth point which we have to consider, that is preparation and insertion of rDNA in the host. Okay, wait a minute. Yeah. Preparation and insertion of recombinant DNA in the host. Okay. Recombinant DNA is prepared, meaning the vector DNA, that is the plasmid DNA, and the alien DNA, they are carrying the genes of interest. Okay. The genes of interest, they are ligated. Okay. And they have now converted into what? The rDNA. Right. So we require two types of enzymes. One is the restriction for digestion and the other one is DNA ligase for joining the complementary sticky ends. Okay. So now the rDNA which is prepared, which contains vector DNA plus foreign DNA, when that is a transformant. Okay. So this rDNA is now it is ready for introduction for insertion of rDNA in the host. Okay. So in the previous we have learned about that how to find out the competent host. Okay. So there are direct or indirect methods to introduce the ligated DNA into the host cells. Okay. There are two types of methods, the direct methods and the indirect methods. The direct methods are the vector mediated methods, and indirect methods are the direct and indirect. Okay. Indirect methods are the vectorless methods. Okay. So the direct and both indirect and indirect methods means the vector mediated and vectorless. Vector mediated and vectorless method. Both are used. Understood? Now, for example, if a recombinant DNA bearing a gene for the resistance to antibiotic ampicillin is transferred into a E. coli cell, okay, ampicillin resistant gene is transferred to E. coli cell. Okay, this ampicillin resistant gene, okay, it is basically the part of rDNA. rDNA. Now, when it is transferred to E. coli, the okay, if such bacteria is transferred to a culture plate, now if the E. coli cell they are transferred to a culture plate having ampicillin, so they cannot grow. They die in ampicillin medium. Okay, so it means the ampicillin resistant gene is not able to produce the protein which will or which is basically it is destroying the ampicillin, or it is making the ampicillin non-functional. So in this way our DNA basically works. So this is the whole picture which is given. Okay, so in this picture I will not start from the beginning because everything is known. Okay, the restriction endonucleases like to cut the plasmid to join, that is the ligation with the help of DNA ligase to seal the gaps. Okay, then this is the nucleoid which is plasmid free bacterial cell, and this is the transformant. Okay, the transformant you can know having the host bacterium with rDNA. Okay, so the transformants they are having a plasmid having the rDNA, then they are multiplied. Okay, multiplication of introduced DNA inside the cell, the clones of DNA are taken out. Now multiplication of the host cells, then clones of genetically engineered bacteria and the fermenter. Now this fermenter is basically bioreactors. We have introduced the rDNA. See, after the preparation, after the synthesis of rDNA, Now we have to check whether this rDNA is compatible to make a protein or not. So we basically want that DNA to be functional, and this DNA will be functional after making protein. Okay, so there are two basic processes. Okay, one is the upstreaming and the other one is downstreaming. Okay, I will write here upstream process, upstream processing. All the steps which are involved in the formation of rDNA. ठीक है, rDNA product. rDNA product means to make rDNA, to make protein from rDNA. So upstream process, it basically involves the formation of products of rDNA, right? Understood? Now the downstream processing. Downstream processing I have explained in the coming slides. Okay, downstream processing. Okay, downstream processing. So before downstream processing, we have to learn about the use of bioreactors or fermenters. I will just write the points what are involved in downstream processing. So downstream processing, it involves the isolation of recombinant product. It involves the isolation of recombinant product. First point. Mean the recombinant product is to be isolated, because if we want to commercialize it, if we want to sell it as a vial, as a ampule, as a medicine, as a drug, as a vaccine, then we have to commercialize it. And if we want to commercialize, we have to purify it, we have to isolate it. Okay, after isolation, then purification. Then it goes for purity testing. Purity testing. Then it is added for preservatives, so that to avoid any secondary infection. If any, to avoid secondary growth of microbes, right? And the last is in the downstream processing, that is packaging. So these are the 5 points in downstream processing. Now, how to obtain the foreign gene product? I mean, how to isolate the foreign gene product? Okay. So isolation of the foreign gene product, to obtain the foreign gene product, okay, we require bioreactors or fermenters. Foreign gene product, basically we need what? A recombinant protein. Okay. So bioreactors, they are mechanical gigantic vessels in which raw materials are biologically converted, are biologically converted into specific products, by microbial degradation, by microbial action, by microbial action. Okay. Understood? Now, these biologically converted products, they can be due to microbial action or plants or animal cells and their enzymes. Animal cells or enzymes. Okay. It helps in the large scale production. And we can produce around maximum 1000 liters, okay, of the product can be processed in bioreactors. Okay, bioreactor, it is of two types: simple stirred bioreactor and sparge stirred bioreactor. There are two types. One is the simple stirred bioreactor and one is the sparge stirred bioreactors. They are basically, you know, difference in the type of blades which are arranged. Bioreactor basically provide an optimal condition for obtaining the desirable product. They provide optimal condition for obtaining the desired product. Okay, understood? Conditions which help to regulate to control the desired product are temperature, pH, substrate, like temperature, pH, concentration of substrate, etc. Okay, now stirring type bioreactors, basically we have stirring type of bioreactors, the most commonly bioreactors which are used. The stirring type bioreactors are of two types, simple and sparged. Okay. Now these simple and sparged bioreactors, they are basically different when they are having different types of blades which are attached. Okay. Now, in the sparge stirred tank bioreactor, sterile air bubbles are sparged. The surface area for oxygen transfer is increased. In case of sparge stirred tank bioreactor, which is free from any germs, okay, sparged stirred bioreactor, it helps to mix, it helps to rotate, it helps to stir, okay, or produce sterile air bubbles. It produces sterile air bubbles, okay. Now, as the sterile air bubbles are produced, the microbes, especially the aerobic colonies, they get good surface area for their action, for their action. Okay, understood? So basically, both the bioreactors, they are based on the fermentation. Okay, bioreactors also known as fermenters. Okay, now types of bioreactors, you can see this is the sparged. Okay, the bubbles dramatically increases the oxygen transfer area. These bubbles, they come up on the surface and they increases the surface area for oxygen transfer. They increase the surface area for oxygen transfer, which further increases the activity of aerobic microorganisms. Activity of aerobes increases. Okay, this is the simple stir type and this is the sparged stir type. Understood? Yeah. Next, The types of fermentation. The fermentation process is a natural process through which microorganisms they turn raw materials into raw materials like glucose, okay, and the other products into alcohol, okay. The fermentation is originally applied only to anaerobic process, so it is originally applied to anaerobic process, okay. But nowadays, previously it was like fermentation was only applied to the anaerobic process. But nowadays, fermentation can be applied both to aerobic as well as anaerobic. Okay, so it can be applied both to aerobic as well as anaerobic. It can be applied to both aerobic and anaerobic process. Now, there is a very important term biomass. Okay, after churning, after the rotation of the blades, you can see the blades when they rotate and they are filled with the vegetation, they are filled with the product, okay. So the product having microbial colonies, the cells, okay, this that is known as what? Biomass, okay. The product is either the cells themselves, biomass, or some useful cell product, okay. Now there is two type of fermentation: batch fermentation, continuous fermentation. In batch fermentation, the nutrients and microorganisms are put in a closed reactor, are put in a closed reactor. They are put in a closed reactor, in a closed vessel, okay, and are not changed from outside once the fermentation start. And they are not changed and not changed after the fermentation starts. Okay, after the fermentation starts, they are not going to change it. Okay, now the example for batch fermentation can be that if we have started the process once, if we have started the process once, for example, there will be no alteration in the concentration of reacting mixture, in the concentration of reacting mixture. Okay, so there will be no change. Okay, so at the last, when the nutrients are utilized, all the nutrients are utilized, the product is separated from microorganisms. Continuous fermentation. In continuous fermentation, the nutrients are replaced as fast as they are used. So we have a continuous supply of fresh biomass, of fresh nutrients or biomass to the reactor. Okay, understood? Removal of waste is also done to avoid spoilage or equilibrium in the biocatalyzed reaction. Is it clear? So in batch fermentation, we don't need to, add anything once we have started the process, we don't need to add anything. But in continuous fermentation, we need to supply continuous nutrients so that fresh nutrients can be consumed and waste can be produced. Okay. Now, the stirred type, that is the first type of the bioreactor, is well suited for large scale production. Stirred type. Which type of stirred type? The simple stirred type. Okay. Or the sparse stirred type. The stirred type, they are helpful in for large scale production. Okay. Under aseptic conditions. Is that clear? It can be used easily in research laboratories, okay, and can be used in research laboratories for research. These state type bioreactors, they can be used to control the oxygen delivery system, okay, foam control system, etc. So batch fermentation and continuous fermentation. Okay. Now coming to next, this is the downstream processing. The downstream process, I have already written in the previous slide that what are the different points. Okay. The downstream process, it involves the intracellular products. The intracellular products, they releases different types of enzymes after the mechanical disruption, physical disruption, chemical or enzymatic disruption. The removal of insoluble and soluble waste comes out, or soluble products comes out, like centrifugation, with the help of centrifugation, flocculation, filtration, or sedimentation. Okay. Then the isolation with the help of adsorption, evaporation, extraction, precipitation, membrane filtration. Then purification with the help of chromatography, precipitation, membrane filtration. Finishing, crystallization, lyophilization, drying, and sterilization. So all these processes, they are involved in downstream processing, okay, to get the final product. Whether we are still on the first step, that is the disruption of cell, then the removal of insolubles, okay, then isolating the insolubles, okay, then purifying it, and then for gone for finishing and polish. Okay. Finishing and polishing. The eukaryotic cells. The eukaryotic cells, the vectors which are commonly used in eukaryotic cells are SV40, simian virus 40. It can accept an insert of length of 4.3 kb length, kilo base pairs, 1000 base pairs. That is, does not contain any selectable marker. Now, there are different types of passenger DNA which we have to discuss. Okay. See, maximum most of the vehicles or vectors are for prokaryotic cells, but you have to remember the names of eukaryotic cells as well. Types of passenger DNA. The types of passenger DNA, it includes first the complementary DNA. Complementary DNA is, it was first synthesized by Temin in Baltimore. Synthesized by Temin in Baltimore. Okay, in the year 1970. In the year 1970. This complementary DNA is basically synthesized on a RNA template. Synthesized on RNA template. Okay, with the help of reverse transcriptase enzyme. with the help of reverse transcriptase enzyme. Okay. Now the DNA is separated from the RNA DNA complex in the presence of alkaline phosphatase enzyme. Separation of DNA from RNA DNA complex by treating it with by treating it with alkaline phosphatase. Okay, by treating with alkaline phosphatase. The complementary DNA strand is formed on the separated single strand DNA template with the help of DNA polymerase enzyme. Okay, the complementary DNA, cDNA, is formed on the separated single strand DNA template on the separated single stranded DNA template with the help of DNA polymerase. Means the first step is first with the help of reverse transcriptase enzyme, the RNA will act as a template, and then with the help of reverse transcript enzyme, a complementary DNA is made or a cDNA is made. The cDNA is treated with alkali so that it can digest the messenger RNA. And then again with DNA polymerase, the complementary DNA strand becomes double stranded. Okay, so it is like that. Previously, I'll make a small diagram over here. Previously, it was sorry. Yeah. We can write it here as well. I can. Yeah. Previously, it was like a single stranded mRNA. Okay, then a single stranded mRNA give rise to single stranded complementary DNA. This is reverse transcription RT. Okay, this red one is the single stranded complementary DNA. I'll make black here. ठीक है? Now, yeah. Okay, like this. So the red one is the single stranded complementary DNA, which is treated with alkaline phosphatase to digest mRNA. Now we get a single stranded DNA template which work as a template. Now it will synthesize what? A double stranded DNA. It will synthesize what? Double stranded DNA with the help of DNA polymerase. So this is the process. Now the synthetic DNA, yeah, that is sDNA. Okay, that is clear. The process of formation of complementary DNA from RNA. Synthetic DNA, it is synthesized on DNA template or without a template. Synthesized on DNA template or without the template. Now, artificial synthesis of DNA on template. Okay. Artificial synthesis of DNA on template means the synthetic DNA. Kornberg. He synthesizes the artificial DNA, okay, on a DNA template only, okay, with the help of DNTPs, deoxyribonucleoside triphosphatases, okay, deoxyribonucleosides triphosphatases, phatases. With the help of this, we can convert a DNA template into a complementary DNA in the presence of DNA polymerase enzyme and some metal ions. Okay. So Correnberg, he take deoxyribonucleoside triphosphatases, virus as a primer, some metal ions. Okay. Then DNA polymerase enzyme. Random DNA in which the small fragments are formed by breaking a chromosome. Fragments are formed by breaking a chromosome, a chromosome of an organism in the presence of restriction endonucleases. Presence of restriction endonucleases. Okay. So this is the synthetic DNA, the complementary DNA, the random DNA. All are different types of passenger DNA or foreign DNA. Okay. Or we can say the DNA insert. The difference between the complementary DNA and the rDNA is that the complementary DNA is obtained from RNA template using the enzyme reverse transcriptase. Please remember, complementary DNA is obtained from RNA template with the help of reverse transcriptase enzyme, with the help of RT. Okay, but while rDNA is, it is a combination of, or it is a making of chimeric DNA or hybrid DNA. Okay, by mixing the genes of two other species. The recombinant products. What are the different recombinant products? The recombinant part, which is synthesize the rDNA. So the mechanical measurement of rDNA or a DNA in cell is in terms of a unit which is known as what? C. 1C. It is a unit, the G1 phase of cell cycle. Okay. The DNA content in 1C human cells is 3.2 picograms. So 1C is the G1 phase of the cell cycle. So if we want to calculate the amount of DNA, like 1C human cells, they have 3.2 picograms of DNA. Okay. Recombinant DNA products, there are different recombinant DNA products like rDNA products are ReoPro. ReoPro is for the prevention of blood clot as an anticoagulant. Prevention of blood clot. Asparag- Asparaginase. Asparaginase is for the treatment of cancers. In the treatment of cancers, especially the leukemia, blood cancer. Herodin. Herodin is also used as an anticoagulant. DNA is to digest DNA. PDGE. PDGE is platelet derived growth factor. Platelet derived growth factor. It stimulates wound healing, helps in wound healing by primary intention. Okay. Then blood clotting factors like factor number 8, factor number 9, anti-hemophilic factor, STOT factor, okay, or Christmas factor. Okay. Interferons, interferons which are used against anti or these are used as antiviral agents. So, my dear students, we have completed the we have completed the process of biotechnology today. In the next turn, we will be starting with the applications. Before that, we will be doing around good number of questions in the next class. Till then, bye bye. Take care. Stay safe. Stay healthy.